Hello, scientists. My name is Dr. Audra Huffmeyer, and I'm a biologist at the University of California, Los Angeles. Today, I'm going to talk about meiosis to understand how gametes, or egg and sperm cells, are produced. At the end of this video, you will understand the differences between meiosis 1 and meiosis 2, the phases of meiosis 1 and meiosis 2, and the products of meiosis 1 and meiosis 2. You will hear over and over in this series that humans have 46 chromosomes and get 50% of their DNA from their mother and the other 50% of their DNA from their father. This is very important in biology and very important in meiosis because this means that sex cells, your sperm and egg cells, have only 23 chromosomes each. So one sperm cell has 23 chromosomes, and one egg cell has 23 chromosomes. And we know that 23 plus 23 equals 46. These are the only cells in your body that do not have 46 chromosomes. The cell division process that produces gametes with 23 chromosomes is called meiosis. Let's learn more about meiosis. Meiosis is a cell division process that reduces the number of chromosomes in sex cells, or gametes, in sexually reproducing animals. Simply, this process takes a cell with 46 chromosomes and produces a cell with 23 chromosomes. Meiosis is very similar to mitosis. Before meiosis, the cell goes through interphase, where it grows and makes copies of its chromosomes. But unlike mitosis, meiosis uses a two-part process, meiosis 1 and meiosis 2. The goal of meiosis 1 is to divide homologous pairs, which is a set of chromosomes, and the goal of meiosis 2 is to separate sister chromatids. In meiosis, cell division occurs twice, okay? One cell can produce 4 egg or sperm cells. Each round of cell divisions, meiosis 1 and meiosis 2, undergo 4 different phases: prophase, metaphase, anaphase, and telophase. During meiosis 1, prophase 1, the chromosome condenses and pairs up with its homologue partner. The partners line up so that they can exchange genes. Crossing over is important for genetic variation among gametes. During the second phase, metaphase 1, the homologous pairs line up in the center of the cell. During anaphase 1, the homologous pairs are pulled apart to opposite sides of the cell. In the final phase, telophase 1, after the chromosomes are pulled to opposite sides of the cell, a nuclear membrane forms around the chromosomes, and the cytoplasm divides, forming 2 daughter cells that are diploid, which means the cells have one set of chromosomes. Now it's time for meiosis 2, the second round of cell division. Cells do not have to prepare for meiosis 2. They do not duplicate their chromosomes. Instead, cells undergo prophase 2, metaphase 2, anaphase 2, and telophase 2 with similar mechanisms as meiosis 1. The results are 4 daughter cells that are haploid, meaning each cell has 1 chromatid. If you are having trouble remembering the order of the 4 phases, P on the MAT, or P on the mat, is a mnemonic device to remember the stages of prophase, metaphase, anaphase, and telophase. Also, remember that while mitosis occurs in all eukaryotes, meiosis only occurs in eukaryotes that reproduce sexually because they need to produce egg and sperm cells. So before we end, I want to check your understanding by doing a quick exercise. What are the differences between meiosis 1 and meiosis 2? Great job. If you listed either of these, you are on the right track. Before meiosis 1, cells duplicate their genetic material, and meiosis 1 results in 2 diploid cells and meiosis II results in 4 haploid cells. To recap, you should now know the 4 phases in meiosis I and II are prophase, metaphase, anaphase, and telophase. Meiosis I results in 2 diploid cells, and meiosis II results in 4 haploid cells. Thank you, scientists, for following along today. I encourage you to keep watching and keep learning.